Okay, ready to dive deep into TGF-beta receptor activation.
Sounds exciting.
It is. We're going to break down this cell signaling pathway, step by step, like from the very beginning to how it changes what a cell does.
You know what's so cool about TGF-beta signaling? It's like it literally determines if a cell is going to grow and thrive
Oh,
or just totally self-destruct, like programmed cell death.
whoa. Okay, so we're talking life or death decisions here. But where do we even start with something this complex? Our listeners sent in some research articles, notes, and I see type I and type 2 receptors mentioned a lot.
Right, those are key players here.
So what are these receptors all about? Like, what do they actually do?
Imagine the cell membrane, right? Like the outer boundary of the cell. Think of it as a city with all these different entry points.
Okay, I'm picturing it. Lots of gates and checkpoints and stuff.
Exactly. And these receptors, type I and type 2, they're like guards at those entry points. They're waiting for a specific signal to show up.
The signal being?
The TGF-beta ligand.
Ah, so the ligand's kind of like the key that unlocks the gate.
Perfect analogy. And the type 2 receptor, that's like the first responder. It grabs onto that TGF-beta ligand when it arrives.
Okay, so it binds to it, then what happens?
That binding sets off a chain reaction. The type 2 receptor, now that it's bound, it pulls in a nearby type I receptor. So now they're like a dynamic duo working together.
Huh, interesting. So it's not just one receptor doing its own thing. It's a team effort.
Totally. And that team, that receptor complex, it's what flips the switch, so to speak.
What do you mean flips the switch? Like what actually happens next?
Okay, so the activated type 2 receptor, it goes on to phosphorylate these proteins called SMAD2 and SMAD3.
Phosphorylate, sorry, just reminding myself. That means it basically energizes them, like gives them a phosphate group.
Exactly. It activates them, gets them ready for action.
SMADs. Yeah, I saw those mentioned all over these research papers. What's so special about them? Why are they important?
Well, think of them like cellular messengers. They carry important info around the cell. And once they're activated by the type 2 receptor, SMA2 and SMAD3, they link up with another SMAD2, SMAD4.
So now we've got like a SMA trio running around.
Yeah. And they've got an important mission. They travel all the way to the cell's nucleus, like the control center of the cell,
Nucleus. And why do they go there?
to bind to DNA, like specific regions of DNA. That's where the real magic happens.
Wait, hold on. So you're telling me these little proteins can actually control which genes get turned on and off?
Yep, that's gene regulation. And depending on which genes are affected, it can make the cell do totally different things.
Like what kinds of things?
Well, it could make the cell divide and grow, or like we were talking about before, it could trigger the whole cell death sequence, apoptosis.
Man, that is wild. So this cascade of events, it all starts with that one ligand binding to the receptor. But then it can ultimately decide whether a cell lives or dies.
Pretty powerful stuff, right?
Definitely. And it makes you realize that even tiny changes in this whole pathway, it could throw everything out of whack.
Absolutely. And that's why when TGF-beta signaling gets messed up, it's linked to all sorts of diseases. Cancer, fibrosis, the list goes on. But before we go down that road,
Yeah.
I think it's worth taking a step back and just appreciating how amazing this signaling pathway is.
I agree. It's seriously mind-blowing how intricate it all is.
Like a perfectly choreographed dance. Every molecule knows its steps and its partners.
Right. But even in the most beautiful dances, there can be missteps.
And when those missteps happen in TGF-beta signaling, that's when things can go wrong, like really wrong.
And the research you sent, it dives into some pretty fascinating examples of that, especially when it comes to cancer.
Oh, absolutely. Like one that really highlights how this pathway can be vulnerable. The story of the VRBA oncogene. It's like this viral hijacker that turns a normal gene into a total rogue agent.
Oncogene, right. I remember seeing that in one of the papers. Something about thyroid hormone receptors.
Yeah. But here's how it connects to TGF-beta signaling.
Okay, lay it on me.
So VRB itself, it's not directly part of the TGF-beta pathway.
Gotcha.
But it shows how messing with the signaling pathway, even in a seemingly small way, can have huge consequences. So VRB, it comes from the gene for the thyroid hormone receptor. And the protein it makes acts like a wrench in the whole system.
Meaning?
It binds to DNA, right? Like it's supposed to. But instead of activating genes, which is what should happen, it blocks the normal thyroid hormone receptor from doing its job. Like imagine a key that fits into a lock, but it can't actually turn it.
So basically jams the system.
Exactly. And that disruption, it messes with how cells grow and develop, which can ultimately lead to cancer.
So it's like a domino effect.
Totally.
Right.
And VRBA, it's like this stark reminder that even tiny changes in these pathways, they can tip the scales towards uncontrolled cell growth, which is like the hallmark of cancer.
Makes sense. But it's not just these viral hijackers that can cause problems, right?
Nope, not at all. Mutations in the genes that actually make up the TGF-beta pathway, those can cause trouble too.
Like mutations in the receptors themselves.
Exactly. Say a mutation makes the receptors lose their tumor-suppressing abilities.
Oh, that sounds bad.
It is. Think of the TGF-beta pathway like a braking system for cell growth. When the receptors are mutated, that braking system fails. Cells can just keep dividing uncontrollably.
And that can lead to tumors.
You got it. It's kind of scary how these small changes at the molecular level can have such a big impact on our health.
It is. So are there other ways that the TGF-beta pathway can get messed up and cause disease?
Definitely. The research you shared, it also talks about the link between TGF-beta and fibrosis.
Fibrosis, right. That's when you get too much scar tissue.
Yep. And it can affect a bunch of different organs, lead to all sorts of health problems.
Not I could.
Nope. So normally, TGF-beta is actually important for healing. Like, it helps make collagen and other stuff that repairs tissue. But when the signaling goes haywire...
It's like the healing process gets stuck.
Exactly. Instead of just patching things up neatly, it keeps making scar tissue, even when it's not needed.
So it's like overdoing it.
Totally. And that excessive scar tissue, that's fibrosis. It messes up the structure and function of the organ. Like imagine trying to build a wall where a window should be.
Yeah, not ideal. It really shows how important balance is in our bodies. Speaking of balance, there's also that connection mentioned between TGF-beta signaling and cyclic AMP signaling.
Oh yeah, that's where things get even more interesting. We're talking about signaling microdomains now.
Okay, I remember reading about something that challenged the traditional view of cyclic AMP.
Right. So the old school thinking was that cyclic AMP just kind of floated around the cell, activating stuff wherever it went. Global signal, they called it. But research on heart muscle cells, it threw a wrench in that idea.
Heart muscle cells.
Yep, cardiomyocytes. They have all these different receptors that can make cyclic AMP when they're activated. But stimulating those different receptors can have totally different results.
Wait, so even though they all lead to cyclic AMP, the effects are different.
Exactly. Which didn't really make sense if cyclic AMP was just this global signal. So researchers had to figure out what was going on.
They didn't answer.
Microdomains.
Okay.
It turns out these signaling pathways aren't just happening randomly throughout the cell. They're organized into these little compartments.
So it's like different signaling zones within the cell.
Yeah. And that compartmentalization, it allows for different things to happen, even when the same molecule, like cyclic AMP, is involved. Think of it like different orchestras playing in separate rooms of a concert hall.
Okay, I'm kind of getting it. Each orchestra makes its own music, even though they might be playing some of the same notes.
Exactly. And the research on kinase anchoring proteins, those are like conductors of those orchestras.
Kinase anchoring proteins, or AKAPs, right?
Yep. They control where protein kinase A goes, which is a key target of cyclic AMP. So they make sure the right signals are being sent to the right places at the right times.
Okay, so it's all about precision and control.
Totally. Got to keep things organized in the cell. Avoid chaos. But how does this all relate back to TGF-beta?
Yeah, that's what I was wondering.
Well, the research we have doesn't directly link them, but it's totally possible that TGF-beta signaling might also be happening in these localized compartments.
So, like, there might be these hidden rooms within the TGF-beta signaling pathway, too.
Maybe. We need more research to know for sure, but even without direct evidence, thinking about these microdomains helps us understand just how complex and precise these signaling networks are.
It definitely does. And this whole deep dive, it's totally changed how I picture what's happening inside a cell.
Me too. It's not just a random jumble of molecules anymore. It's like this incredibly organized and dynamic system.
It's wild to think about all this happening inside of us right now in every single cell. We've uncovered a lot about how TGF-beta receptors get activated, from that first ligand binding to how it affects which genes get expressed.
And like we've been saying, if even one tiny thing goes wrong in that whole process, the consequences can be huge, like with cancer and fibrosis.
Yeah, definitely not good. But learning all this about these mechanisms, it makes you wonder, right?
What do you mean?
Well, like, if we could actually manipulate these pathways, maybe we could come up with new treatments for these diseases.
That's exactly what researchers are trying to do. Imagine being able to tweak the TGF-beta pathway. turning it up or down as needed to help heal wounds, stop tumors from growing, even regenerate tissue.
It sounds almost too good to be true.
Yeah.
But then again, it is based on all this amazing science, years and years of research going into these pathways.
And we're still just scratching the surface. There's a ton more to learn about TGF-beta signaling, how it interacts with other pathways, all the little things that control it. But what we do know so far, it's really valuable.
Totally agree. I feel like after this deep dive, I have a whole new understanding of how complex and just elegant these cellular processes are.
Me too. It really shows you the power of science, and how it can help us improve human health.
Absolutely. So as we wrap up our look at TGF-beta receptor activation, what are some key things you'd want our listener to walk away with?
I think the biggest thing is that even though these pathways are happening on a microscopic level, they have huge effects on us. They affect our development, our health, even what diseases we might get. Understanding them is like a key part of figuring out how our bodies work.
Super important. And it just goes to show why research in this area is so crucial. Who knows what other discoveries are waiting to be made?
Exactly. With every new finding, we get closer to developing new treatments and therapies that could totally change how we approach healthcare.
Well, that about wraps up our deep dive into TGF-beta receptor signaling. It's been an awesome journey. I hope you, our listener, have learned a lot about how incredibly intricate your cells are.
And remember, this is just the start. There's so much more to discover about the amazing world inside our cells. Keep those questions coming, stay curious, and you never know. Maybe you'll be the one to make the next big breakthrough.
Until next time, keep exploring.